and thank you for joining us today. My name is Stephanie White,
Malika Ranjini, Itrit Batoon,
Isaac Hernandez,
and we worked on the project Blood Procoagulant. We did this under the mentorship of Damien Padella, our faculty advisor, Dr. Galen Stuckey, and with the help of our resident assistant, Jenny Lay. So what do you think is the third leading cause of death for all ages? It's the leading cause of death for ages 45 and under, and accounts for 30% of the deaths per year in this age group alone. Maybe heart disease, cancer, AIDS? Well, it's actually traumatic accidents. So in a traumatic accident, your body can go into a state of shock. This can be brought on by many different physical conditions, one of which is hypothermia. So when a person is hypothermic, their body's core temperature, so the temperature in their abdomen, where all their important bits are, drops below 35 degrees Celsius. At these cold temperatures, your body's natural processes slow or even stop. Now imagine this, coupled with severe blood loss. These are the conditions we tried to mimic in the design of our experiment. So how does our body naturally try to stop these conditions?
Imagine our body is like an airtight machine, so if it gets a cut somewhere, all the blood will eventually come out and we'll die. But this doesn't happen. It's because of the clotting process, also known as coagulation. It's a highly complicated process. There are lots of factors involved in it, but this is really a simplified version of how it works. Prothrombin is a protein. It is also a clotting factor that is present in our blood. It is in an inactive form, but when there is an injury, tissue factor activates it into thrombin. Thrombin then triggers the conversion of fibrinogen into fibrin. Fibrin is essentially a mesh of protein network that covers the damaged area until the tissue underneath it is healed and the wound is thus, Calcium pushes each step up to the production of thrombin. They all work together and they set off the activation for the other factor. As you can see, prothrombin is the main factor that is involved in the clotting cascade. So one of our project goals was to determine the activity of prothrombin on the formation in the clot formation. We also dealt with different concentrations of prothrombin at different body temperatures. We basically focused on hypothermic temperatures that was like 35 degrees centigrade and below.
Tromboelastography is also known as TAG, is a method we use to test how long it takes for a liquid blood sample to convert into a solid clot. How TAG does this? We can use TAG to at different temperatures. And like Itara just mentioned, we were mainly focusing on hypothermic. So how this works? We insert a small blood sample you guys can't see it, but in here is the pre-warmed, and this is what heats the blood to a desired temperature. We insert a small blood sample, then we drop this pin. This pin is a sensor that slightly rotates from side to side. As it rotates from side to side, when it's liquid, there's no resistance. However, when the first sign of clot, it puts resistance on the movement of our sensor. Now, all this data is transformed to the screen, and here this is the graph that pops up at our screen, and it models exactly what's the activity which is going on. And the data that the sensor was capturing. From TAG, we can gather a lot of data in terms of the size of the clot and the strength of the clot. However, our main focus was this region. As you can see, as I mentioned, when it's liquid, there's a straight line. And when the first clot, when it senses the clot, this red line is what tells us that there's a clot. So we tested several. We tested one trial at several concentrations. We tested at 1 milligram per millimeter, as well as 5, 10, and 20. And our main focus was to find the best concentration in which the clot quickly begins to form. And we found that it was on 10 and at 20. So, however, due to lack of time, we were only able to run these tests once. However, to further support our data, we used another instrument called the plate reader.
Now, what the plate reader does is that inside this microplate, we're mixing the prothrombin and the plasma with a factor of fluorescent dye and calcium. As Itra previously mentioned, the calcium is really an essential part of forming the clot. So as you mix all of these different components, the clot starts to form in this little microplate. But inside the device, it's completely dark. So we would want to excite the fluorescence to be able to measure how much clot is shaping out each time. So we shed light on the microplate and excite the fluorescent dye on the surface area of the clot, and then measure the fluorescence. Now, as the clot is growing, we're going to have more surface area, and therefore more fluorescence will be emitted. So we will have a larger amount of fluorescence on our diagram. So as you can see in this diagram, the more, as it's shown right here, the more concentration of prothrombin we have, like the blue diagram, forms the maximum amount of clot faster, so at a sooner time. And then after that is the 10 milligram, and then the 1 and the 5. Now, the previous experiment was done at 32 degrees, but this one was done at 35 degrees. And as you can see, the whole slope thing isn't really working out. It's just like all over the place. So why is that? Because all the graphs are supposed to be like pretty similar. Now we kind of examined what we did differently in this experiment, and we figured out that we froze the dye before use. So next time you're experimenting with human blood, don't freeze your dye, and kids don't try this at home.
So upon examining the data, we noticed a possible trend. And this actually occurred at 32 degrees Celsius, and it occurred between both results of the machines. So we look here at the TEG, and at this 5 milligram per milliliter concentration here. As we can see, it took the longest time to form the clot. And so we actually looked at the plate reader to see if that was also another result there. And as you can see from this green line, when it reached this maximum point, it took much longer than the other concentrations. And so due to our limited data, we were not able to find an optimum concentration, but with trends like these between the two machines, we can hope to someday discover one. Okay. So another thing, as Malika mentioned, is we learned definitely that we have to make the dye fresh. And so we will coin the term from Jack in the Box, we won't make it till you order it.
So with our research project, like right now we have been working on external forms of treatment, but we can definitely apply the knowledge to development of intravenous forms of treatment, which will help the hemorrhage patients that suffer from internal bleeding, and will be really beneficial with future research and future further. Working on this project has been a great learning opportunity. We would like to thank you, our mentor, Damian Fidela, Professor Gillen Sticky, in charge of the lab we worked in, UCSD faculty and staff, JKC Foundation for funding, staff and all the resident advisors for all their guidance and support. Thank you.